The overall goal of this procedure is to manufacture and characterize polymeric thin-film composite structures for advanced cell culture studies or as skin adhesives.
This method can help answer key questions about how elastomeric composites can be used to achieve optimal sticking action, even on irregular surfaces. The main advantages of these techniques are that they are simple and universally applicable. This allows large-scale manufacturing with a very high precision.
Generally, individuals new to this method will struggle because the adhesion measurements require intensive training and practice to achieve the necessary precision.
Demonstrating the cell culture procedures and plasma application will be Angela Rutz, a technician from our lab.
To prepare the PDMS, mix and degas 1.1 grams of the pre-polymer mixture in a speed mixer at 2,350 RPM under vacuum for 3 minutes. To prepare the PDMS backing layer, use an automatically controlled Dr. Blade application machine. Place the Dr. Blade on top of a 100% isopropanol cleaned piece of PET foil and use the micro-positioning screws to adjust the thickness of the backing layer. Load the PDMS into a single-use 10 milliliter syringe. Fill the reservoir of the Dr. Blade with the polymer and start the movement of the blade with a velocity of approximately 2 millimeters per second. When the piece of PET film has been coated, transfer the film into a 95 degrees Celsius oven for 1 hour.